My name is Vincent, so we're going to talk about today about experimental design, which could be a potential question in a year 11 exam. So the question context is like this, so if we have a look, and basically it's trying to say the strawberry can be prepared and preserved in different ways. So from this table, it gives us an important hint of what is meant by strawberry preservation. So the preservation on the other alternative side, it basically also means it's been colonized or eaten by microbial organisms. And these microbial organisms could be bacteria, fungus, or anything that are eating the bacteria other than us. So therefore, to limiting the bacterial growth will be paramount important in terms of preserving the strawberries. Okay, so in the sense that if we're looking at these question context here, there's a table you need to fill up. Basically, it's been given with some preservation techniques, either cooked or dried, and explain how it's going to limit the bacterial growth and eventually the shelf life extension. What do you expect? Is it going to be longer shelf life or shorter shelf life? So in a cooked way, so we're basically using temperature to achieving sterility or achieving disinfection for these food contents, which is the strawberry. So by cooking them, we're denaturing the enzyme, destroying their nucleic acids, and bioorganism doesn't have an intact nucleic acid, nor or nature enzyme is going to be pretty hard to live on its life and replicate. So therefore, it limits the growth of or replication of microbial organisms. And so therefore, in a sense, we would expect the shelf life to be longer. And dried technique is basically by depleting the free water from the food contents, so therefore limiting the water accessibility for those bacterial organisms, and therefore we also would expect the shelf life to be kind of long, but in comparison with these two, we have no way of knowing which way is going to be the best thing to do. So this table seemingly a little bit nonsensical, but really it is providing a logical extrapolative path for you to pursue later in experimental design. So in the second part of the question, it's going to tell you a story, non-relevant. So you don't really have to go through the story if you don't want to, but really the important bit is right there. So it's which method of the preservation will give the strawberry the longest shelf life. So we talk about cooked versus dried, and it could be something that you're starting with, but I'm not going to start on these two preservation techniques today. I'm going to starting on what's the temperature we're cooking them will, or what's the temperature we're storing them or treating them will achieving a longer shelf life. So, alright, so this is basically the experimental framework, the design framework that we're going to go through today. The first thing to consider, as the question was actually asking, and it's basically what are the factors that are important in affecting these steps. So if we consider what factors must be control variable, so that could include the pressure, the pH of the water we're adding in, and the amount of water we're adding in, and also the amount of inoculating volume. We're going to talk about these in the end, because it would not make any sense if we're given with a particular experimental setup. So the setup is very easy. We could have started with 50 strawberries for each of the group, minus 28 RT and 100 degrees Celsius. And we blend them. Why do we need to blend them? I'll talk about it in the end. So if we don't blend them, that's fine. We can just freeze it for 100 minutes, RT for 100 minutes, and 100 degrees Celsius for 100 minutes. And that will be treated as identical conditions, but however, the only thing different is temperature. So after 100 minutes, we took them back into RT and have a look at food spoilage. But food spoilage is a concept and it's not really quantifiable, it's not really measurable. So we must translate that concept into something can be measured. Now, We said earlier about food spoilage or the other alternative side of food spoilage is being occupied or colonized by microbial organisms. So therefore, we need to find a way to measure the amount of microbial organisms. So we can do this by blending these into juice to start with, and taking a subfraction, say, well, I don't know, maybe 50 microliters, to inoculate our nutrient agar. And these nutrient agar will, of course, be inoculated from -20 room temperature and 100 degrees Celsius. And then we're going to incubate these nutrient agar plates, which has inoculated by this juice overnight at 37 degrees Celsius. And on the next day, we count the number of colonies that are formed. So we might have been expecting in the room temperature, which is our comparison point, had a certain number of colonies that are starting to form. And the frozen condition, we also had a few other colony numbers to form, and 100 degrees Celsius, we had less number of colonies to form. So in a way that we can plot them into a bar plot as our expectations. It's not the real data, but it's our expectation. We would expect the room temperature to be the comparison point, and stuff we did as freezing as a lower number of colonies, and boiling has the lowest number of colonies too in the end. So the other question is, how do we be sure that these colony number differences is not happening by chance? It is something systematically different. So we can only be sure by increasing the number of replicates in the whole experiment. Instead of having just one group independently, we can have 3 independent groups. So I'm repeating that by 3 times, that by 3 times, and that by 3 times. So for this, all of the design will then be in a real framework, and you don't really have to consider the factor first if you don't want to, and if you have the design in your mind, and when going back to consider what are the factors that are really important in terms of reaching a conclusive answer, is by controlling other environmental factors to be the same. So pressure, we talked about, pH and the volume that we add, volume of water adding to these jars to make that juice, and also the amount of volume that we're taking from this juice to inoculate our nutrient agar plates. Okay, I think that'll be really the end of it.